Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper for the subject Life Science, of course. It is a shift to part C questions. It was conducted in July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR Net JRF being discussed, you can go to my channel Omni Bioentrance, select CSIR JRF Net in the playlist, and there you will find. Now, the phylogenetic tree given below shows single nucleotide polymorphism observed among 4 individuals of the scorpion species. So, these are the various polymorphism. Select the option that represent the correct combination of ancestral nucleotides at nodes X, Y and Z using the principle of parsimony. So here if you see, Y has got the nucleotide A. Y has got nucleotide A, X has got nucleotide A, Z has got T and A. So that is seen in option 3, YA, XA, ZA or T. So option 3 is the answer, and that is what they have asked in the question. Then next one. The following tree shows phylogenetic relationship between species A and D. So this is the species A, B, C and D. Same in this branch also, species A, B, C and D. So which of the following molecular mechanism would be responsible for the phylogenetic relationship shown between species A to D? It is just gene duplication. Then next one. The mutation rates refer to the frequency at which new mutations arise in the genome of an organism and is typically expressed as mutation rate equal number of observed mutation by total number of opportunities for mutation. Which of the following factors will not influence the opportunities for mutation? Generation time. Yes, species with shorter generation time have higher mutation rate, so that will influence. DNA repair efficiency and replication fidelity. If DNA repair genes are inactivated, mutation rate increases. Exposure to mutagens again increases mutation. But population size, it will not influence the opportunities for mutation, because sometimes large populations can tolerate higher mutation rates. So the answer here is option 4, that is population size. Then following statements are made about for the production of cisgenic plants. So what are these cisgenic plants? They are genetically modified plants that contain genes either from the same species or from sexually compatible donor plants. What is the difference between cisgenic and transgenic? Cisgenic, the recipient plant or the receiving plant, the plant which receives the gene, it will get these modified genes from non-plant organisms or from a donor plant that is not at all related in any way to the recipient plant. It is sexually incompatible recipient plant. So that is transgenic. Whereas in cisgenic, they contain genes from the same species or from sexually compatible donor plants. Okay. Now, first statement. In cisgenics, the donor sequence does not necessarily replace the native gene sequence but is added to the recipient species. That is correct. Because cisgenic, I told you, it is a genetic modification technique and involves transferring of a gene from donor plant to a recipient plant without replacing the native gene. The donor and recipient plants are from species that are sexually compatible or they are closely related. So A is correct. Cisgenic plants might contain DNA sequence such as tDNA borders from the plasmid vector. Yes, if cisgenic plants are produced through agrobacterium mediated transformation procedures, then tDNA borders are sure to be found. Insertion of a cis gene may result in a gene mutation at the site of insertion similar to that of transgenics. Yes, that is also correct. With regard to the species gene pool, cisgenic does not alter the gene pool of the recipient species. That is also correct. Because in cisgenics, it involves introduction of genes from a sexually compatible species. So it does not change the gene pool of the recipient species. It merely adds to it without introducing foreign genes from unrelated species. So D is also correct. Both cisgenic and transgenic can use the same DNA transformation methods to introduce the respective gene construct into the recipient plant. That is also correct. It can be either through agrobacterium mediated transformation, biolistics, etc. So here all A, B, C, D, E are correct. Then next one. Regulatory RNA mechanism for the control of gene expression. Ribose switch. Ribose switch is change their conformation when bound to small molecules, usually metabolites. A2. A2 is there only in option 1. So that must be your answer. MicroRNA. B1. Base pairing with specific mRNAs and controlling their stability and their translation. B1 is correct. Small interfering RNA, complementary base pairing followed by RISC mediated mRNA cleavage. C3. So A2, B1, C3. 1 is correct. Then next one. A receptor tyrosine kinase dimerizes and autophosphorylates in the presence of a ligand. Okay. RTK, receptor tyrosine kinase, it will dimerize, it will autophosphorylate when it is bound to a ligand. That is correct. A researcher prepared 3 constructs that express either the A, full-length protein having a kinase domain as well as 3 tyrosine residues. That is in case of A. In B, the RTK is non-functional kinase but 3 tyrosine residues are present. Whereas C, the RTK lacking 3 tyrosine residues but having a functional kinase. So this RTK, it phosphorylates at the tyrosine residues. Okay. That is why these constructs are made with RTK as well as tyrosine residues. She expressed these constructs in cell lines lacking the RTK, either singly or in combination shown in the figure. So either A, B, C was single or A plus B plus A plus C, A plus or B plus C combinations were used and expressed the constructs in cell lines. Then breaks open the cell and add the ligand of the RTK in the presence of radio-labeled ATP. So with the help of ATP, she can phosphorylate the RTK. She immunoprecipitated the RTK and analyzed the immunoprecipitate by Kumasi staining as shown in the figure, followed by autoradiography. Which of the following autoradiograms would the researcher expect? So if it is A, you will get a band here. If it is B, you will get a band here. If it is C, you will get band here. If it is A plus B, you will get these 2 bands. If A plus C, these 2 bands. If B plus C, these 2 bands. Now see what is happening. Which one of the following autoradiograms would the researcher expect? So A is having tyrosine kinase domain plus 3 tyrosine residues. So there is region for phosphorylation. RT kinase is perfectly working. So it will phosphorylate. You will get a autoradiogram. So A will give a band over here. So A band is coming here. So in this autoradiogram also A band is coming here. So in every options A is having a band. B doesn't have functional kinase, but it has 3 tyrosine residues. But without kinase, what will autophosphorylate the tyrosine residues? Nothing. So B will not give a band. So in all cases, B is not giving a band. 3, it is lacking the tyrosine residues but has functional kinase. So 3 tyrosine residues are there, but kinase is not there to phosphorylate them. So it will not give a band. So C is also not giving band in all the options. Now coming to combinations, A plus B. A plus B. Now A is having functional tyrosine kinase, B is not having the tyrosine kinase but it has 3 tyrosine residues. So A will give a band because it is having functional kinase and 3 tyrosine residues. Now A's protein kinase itself will phosphorylate the 3 tyrosine residues of B. So B will also give a band. So you will get 2 bands in A plus B. You get 2 bands here. In option 2 also you get, option 3 also you get, option 4 also you get 2 bands. Now A plus C. A plus C, A is having protein kinase domain as well as 3 tyrosine residues. But C, it is lacking the 3 tyrosine residues but has functional kinase. So that case A band only you will get because A is having kinase domain as well as 3 tyrosine residues. But C, it is lacking 3 tyrosine residues, so no region to phosphorylate. So C's band will not be present in A plus C combination. Only A's band will be present. So first one A band is present, second one C band is present, so it is not correct. Third one A and C are present, so it is not correct. Fourth one A and C are present, so that is also not correct. So your answer here is option 1. Now see B and C. B plus C, B has the non-functional kinase and 3 tyrosine residues. C is lacking the 3 tyrosine residues but having functional kinase. So the functional kinase of C will phosphorylate the 3 tyrosine residues of B, giving a B band in the autoradiograph. But C band will not be present because it is lacking the tyrosine residue. So no place for the kinase to phosphorylate. So B band will be observed, but C band will not be observed. So 1 is your answer. Then a region of eukaryotic chromosome is heavily transcribed by RNA polymerase II. Given below are few properties of such a chromatin. DNase 1 hypersensitivity. Yes, DNase 1 hypersensitivity region is a region of the chromatin that are sensitive to the cleavage by DNase 1. And this is functionally related to increase in the transcriptional activity. So a region is heavily transcribed by RNA polymerase II. So this region will be transcribed by RNA polymerase. So A is correct. A high CpG methylation. Now methylation of CpG in the gene or in the promoter region of the gene can silence the transcription. So B is not going to be correct. It is not a highly transcribed by RNA polymerase region. Then occupied by macro H2A. Macro H2A is a histone variant. It is a repressor of transcription. Okay. So the region occupied by the chromatin by macro H2A will not help in transcription. So C is also not correct. Then high histone acetylation. Yes, it is associated with transcriptional activation. Histone acetylation, it will loosen the chromatin structure, allowing transcription factors to bind there. Okay, so A and D are correct. Answer is option 4. Then next one. A decapeptide composed of MFTGPYCRPW. Okay. Was dissolved in 20 millimolar Heps at pH 7, 50 millimolar NaCl, 50 millimolar Na2SO4, 5 millimolar DTT and 4 millimolar EDTA. Which one of the following statements about the peptide in the given buffer condition is correct? Buffer condition means Heps buffer, that is at pH 7. What is the correct statement? The peptide forms a dimer through disulfide bond. Now the peptide, if you see, it is having only one cysteine residue over here. There are no cysteine residues there. How will it form disulfide bonds? For disulfide bond formation, you need 2 cysteine residues. But that is not there. So first one is not correct. The peptide has a net positive charge. That is correct because arginine is present here and at pH 7 arginine is having a net positive charge. Others are not charged groups. Okay. The peptide has net negative charge. That is also not correct. The peptide is neutral. That is also not correct. So 2 is the answer. Then next one. Protein A binds to the mRNA for gene B in HeLa cells. So protein A binds to an mRNA. Now this is the mRNA. Protein A binds there to the mRNA. The protein A mediates the formation of mRNA protein particles, mRNPs. So this is mRNP. The addition of a chemical C disrupts mRNPs in HeLa cells. The result of the Western blot and Northern blot analysis are shown below. So this is Western blot for A, this is Western blot for B. So these are the HeLa cells, these are HeLa cells treated with C, HeLa cells and HeLa cells treated with C. This is Northern blot pull down with A. Probe for B, HeLa cells and HeLa cells treated with C. GAPDH is just a control. Okay. Now if you see there here HeLa cells treated with C means the addition of chemical C disrupts mRNPs in the HeLa cell, but here it doesn't disrupt. Even though even if mRNPs are disrupted, it does not affect the interaction of protein A to the transcript B. Okay. Here also, even if they treated with HeLa cells, Western blot for B, if you see, treat HeLa cells with C, there is band obtained. So it is not disrupting the interaction. Here for mRNAs, HeLa cells treated with C, again there is no disruption of the band. Treated with C, band is there. So from the above experiment, which of the following statement is true? mRNP disruption does not affect the interaction of protein A and transcript B. Yes, that is why you got bands everywhere. Or the translation of transcript B. Or translation of transcript B, that is why you got a band over here. It is getting translated. Protein A is interacting with B and it is translating gene B. That is why you got a band over here also. So first statement itself is correct. Then following are different critical reaction steps involved in the oxidation of lipids in many organisms. Okay, this is beta oxidation. The correct sequence they have asked of the steps. So first one will be hydrolysis of thioacyl glycerols by lipase. This is first one. Then comes activation of fatty acid by conjugating to CoA. This is second one. Then comes reaction of fatty acyl CoA with carnitine. That is 3. Then comes hydration and then thiolysis. So the order will be C, D, A, E, B. C, D, A, E, B is there in option 1. So option 1 is the answer. Then subcellular compartments with their markers you have to match. Endoplasmic reticulum, it is protein disulfide isomerase. So A3. Okay, A3 means 2 and 4 gone. Golgi apparatus, it is monosidase 2. B4. B4. In 1 and 3, B4 is there. Now autophagosome, it is LC3B, C1. C1 is there only in option 1, so option 1 is your answer. Mitochondria, HSP60, D2. So the answer is A3, B4, C1, D2.